Hello everybody, today I'm going to talk about muscle contraction. This is my third lesson presented by Sally Ahmed Khalil. So here we have three types of muscles in our body. We have skeletal muscle, cardiac muscle and smooth muscle. And the name of muscle comes from a Latin word which is musculus, which means little mouse. We have a skeletal muscle is also known as voluntary muscle. Why voluntary? Because I control my movement. So when I decide to raise my hand, this is voluntary because I decide to do this action. Okay, and here cardiac muscle is involuntary. Why? Because I can't control the beating of my heart. Okay, with my heartbeat, it beats automatically and I can't control its beating. And we have here smooth muscle, which is like uterus, urinary bladder, and the stomach. They are also contracting involuntary because I can't control their contraction or movement. So we have here approximately 650 muscle attached to our bone, so in our body. So here skeletal muscle is also known as voluntary muscle and is also known as created muscle. So why it is named skeletal muscle? Because these muscles are attached to the skeletal system, as you see here. This is my skeletal system. And here voluntary muscle, why? Because when I want to raise my hand, for example, I send signals to the brain, and this brain will send these signals through the nerve. It will pass down here until it reaches the muscle, okay, at the end here, until it will reach the muscle, and then it will give the order to contract, so you could then be able to raise your hand. Why it is named striated? Because here we have the unit of muscle contraction is the sarcomere. Here it is bordered with two Z line, as you see here. Okay, and here is actin filament, which is a thin filament, is attached to the Z line, and here is a thick filament, which is called myosin filament. It is attached to the M line, and here they are the actin and myosin filament are arranged here in a striated form. From here the name striated came. We have here muscle contraction. So it happens through a sliding filament mechanism, which I will talk about it later. This lady here, she has a muscle here, okay, and her muscle consists of a bundle of muscle fibers, okay. So here this is a muscle, and here it contains a bundle of muscle fibers. These muscle fibers here, there's an extension of them, and you know here there is, you could see here, numerous nucleus, and each muscle fiber consists of numerous number of myofibril. This is an extended myofibril at the end here, which contains the unit of muscle contraction, which is a sarcomere, and this is a board. It is bordered. Each sarcomere is bordered with two Z line, as you see here. So here is the magnification of this Z line or sarcomere. So you could see here, this is an electron microscope of the sarcomere, where you could see it is exactly the same. So here you could find this is the actin filament attached to the Z line. This is a myosin filament and thick filament attached to the M line. And here is the A band, which is only consisting of myosin. And here is the I band, which consists only of actin filament. And here the A band, it is an overlapping area between actin and myosin. You could see it is a dark area here. This is the A band, okay, and this is the whole sarcomere. So here there are some definitions that I would like you to know. What is ATP? It is adenosine triphosphate, which is the source of energy in our body. So adenosine triphosphate, when it is hydrolyzed, it gives ADP, which is adenosine diphosphate plus phosphorus. We have action potential. Action potential happens when there is depolarization happens in the nerve, in the membrane of the nerve, and it passes through the nerve through one direction only. Okay, I talked about action potential in my previous lesson. We have sarcoplasmic reticulum. It is the endoplasmic reticulum, but within the muscle. So the endoplasmic reticulum is a source where we could store the calcium. So this is a place where calcium is stored. So sarcoplasmic reticulum, you could find calcium there. Acetylcholine is a neurotransmitter located at the end of the nerve, and these are present in synaptic vesicles. When an action potential passes through the nerve, then it stimulates the release of those acetylcholine. Then they go to the receptor of the muscle and stimulate another action potential to happen, and then stimulate the release of calcium from the sarcoplasmic reticulum, and the process of muscle contraction will start. We will know that how it is happening here through the sliding filament mechanism. Here is the muscle contraction, where we could find here, this is the contracted form of the sarcomere. It is shorter than the relaxed form here, as you see here. Okay, so here we have actin filament, which is a thin filament. It consists of tropomyosin, which is a thread here, and it consists of troponin. When calcium is released from the sarcoplasmic reticulum, then it goes and attaches the troponin, and then it will pull the tropomyosin away, so it will expose this myosin binding site, will be exposed to be able to be attached to the head of the myosin later. Okay, and then here we could find that ATP, when it is attached to the head it got detached, and when it is hydrolyzed to give ADP and phosphorus, then the head is exposed here to be attached to this myosin binding site, and okay, to form through a cross bridging mechanism. And here this head will then move inward when there is releasing of ADP and phosphorus from it. Okay, so the head will move inward towards the M line, and then when another ATP is attached to, so the head got relaxed again, and this process continues again and again until the muscle is going here contracted. The sarcomere will be contracted, okay, and here the actin filament being moving toward the M line, okay, so it becomes shortened, as you see. So here is a summary of what I said. Here, when you got a signal passing through the motor neuron, so then they will go to this muscle, and then here we have a synaptic vesicle which contains acetylcholine on inside them. When they got an action potential, then that is responsible for releasing those acetylcholine to be attached to a receptor here on the membrane of the muscle, which is the sarcolemma. And this membrane here then will pass this action potential in the T-tubule, and this T-tubule will stimulate the release of calcium from the endoplasmic reticulum. And when calcium is released, as you see here, it is attached to troponin, and then it will pull the tropomyosin away, so the myosin binding site will be exposed to the head here, the myosin head. And then when myosin head is at, it has each ATP is hydrolyzed to give ATP and phosphorus, then the process of movement happens. So here is a relaxed form of the muscle, and here is a contracted form of the muscle. Okay, and here is a sarcoplasmic reticulum, and here is a T-tubules where the action potential passes through. Okay, here is how to communicate with me. You could communicate by asking questions or leaving a comment, or by also answering my questions here through my YouTube channel, Learn With Me, and through my Twitter account, and also through WordPress. Here are my references that I used through this presentation, and here are the questions that I would like you to answer, please, and you could send them to me through Twitter or my WordPress as a blog or mini blog. Thank you very much. I would also appreciate if you could leave a feedback and comment, and you put a thumb up if you like this video. Thank you very much. And now I would like you to watch a video. It is a 3D video. I got it from the YouTube. Okay, so I put the start of this video on my reference, and I would like you to watch it because it will facilitate and let you understand more what I was talking about. So thank you very much and enjoy.
You use muscles every day to do activities. This woman is using muscles to breathe, circulate blood, and move her hand to take notes. Your cardiac and smooth muscle tissues are involuntary. You do not consciously control their actions. Skeletal muscle works under voluntary control. Skeletal muscles are composed of bundles of muscle fibers. Muscle fibers are long cylindrical cells containing several nuclei. Muscles will contract or relax when they receive signals from the nervous system. A neuromuscular junction is the site of the signal exchange. This is where the synaptic bulb of an axon terminal and muscle fiber connect. Muscle fibers are composed of many myofibrils. A myofibril contains contractile units called sarcomeres. Sarcomeres run adjacent to one another down the length of the myofibril. Each sarcomere consists of alternating thick and thin protein filaments, giving skeletal muscle its striated appearance. The muscle contracts when these filaments slide past each other. The thick filaments are myosin, which are anchored at the center of the sarcomere, called the M-line. The thin filaments are composed of the protein actin, which are anchored to the Z-lines on the outer edges of the sarcomere. Because the actin filaments are anchored to the Z-lines, the sarcomere shortens from both sides when actin filaments slide along the myosin filaments. Although the action between the filaments is described as sliding, the myosin filament actually pulls the actin along its length. The cross bridges of the myosin filaments attach to the actin filaments and exert force on them to move. This action is known as the sliding filament mechanism of muscle contraction. In this model, the sarcomeres shorten without the thick or thin filaments changing in length. A contraction begins when a bound ATP is hydrolyzed to ADP and inorganic phosphate. This causes the myosin head to extend and can attach to a binding site on actin, forming a cross bridge. An action called the power stroke is triggered, allowing myosin to pull the actin filament toward the M-line, thereby shortening the sarcomere. ADP and inorganic phosphate are released during the power stroke. The myosin remains attached to actin until a new molecule of ATP binds, freeing the myosin to either go through another cycle of binding and more contraction, or remain unattached to allow the muscle to relax. Muscle contractions are controlled by the actions of calcium. The thin actin filaments are associated with regulatory proteins called troponin and tropomyosin. When a muscle is relaxed, tropomyosin blocks the cross-bridge binding sites on actin. When calcium ion levels are high enough and ATP is present, calcium ions bind to the troponin, which displaces tropomyosin, exposing the myosin binding sites on actin. This allows myosin to attach to a binding site on actin, forming a cross bridge. Calcium ions are stored in the sarcoplasmic reticulum and are released in response to signals from the nervous system to contract. Neurotransmitter molecules are released from a neuron and bind to receptors, which depolarizes the membrane of the muscle fiber. The electrical impulse travels down the T tubules and opens calcium stores. Calcium ions flow to the myofibrils, where they trigger a muscle contraction. As the actin and myosin slide along each other, the entire sarcomere shortens as the Z-lines draw closer to the M-line. As the sarcomeres in myofibrils contract, the entire muscle fiber will shorten. When muscle fibers contract in unison, a muscle can produce enough force to move the body, allowing you to take notes.
So thank you very much for watching, and don't hesitate to ask any question, please. Thank you.